Good afternoon, everyone, and welcome to today's webinar with Qlucore. You are here with me, Mats Svensson, who is your host today. And normally I work with development here at Qlucore. I also work with support and doing these webinars, so some of you who have been in contact with support might have run into me before. Today's webinar is going to be a very, very simple introduction to some of our plots and what you can do with our software. And I'll start with a very short presentation of Qlucore as a company and what we're doing. Our main product, Qlucore Omics Explorer, is a very fast and easy to use tool that you can use to analyze and explore data without being a statistical expert. That's the whole point. The tool is tailored towards those who want to do exploratory analysis on their data. They want to dig into their data and find out more about what kind of complex information is in there. Also, Qlucore Omics Explorer is different than many other software packages and offerings out there because it's instantaneous. It's extremely fast. It's interactive. So you actually interact with your data and you see your data on the screen while you are performing analysis on it. Also, it works on normal computers. You don't have to run this on a very, very fast computer. You don't have to run this on a cloud server or something. We offer this for PC and Mac, and you can run it on a regular laptop, actually. Now, the background here to Qlucore as a company. We started off, or what became the company started off as a research collaboration about 20 years ago in 2001 between mathematical science and clinical genetics at the Lund University, where the researchers at genetics suddenly had lots of interesting data, but they didn't know what to do with it because it was too much data. They had array data from array studies, but that was just bunch of tables that didn't make sense to them. At the same time, mathematical sciences had started doing research into what became known as big data later on. And these methods were combined with the data from clinical genetics. And we ended up with, in 2007, what became the Qlucore company. We were founded in 2007 in Lund in Sweden. Now we have customers on all populated continents, I should say, because Antarctica isn't considered populated. So I don't know of any customers in Antarctica anyway. Maybe some research station there runs it. Who knows? Anyway, we have customers around the world. We have sales offices in Sweden and the United States, and we cover most of the big universities and pharma companies out there. Here's a few examples of customers of ours. But that's not the whole point here today. The whole point here today is to show you the software. I just want to mention also that we on our homepage have publications and case studies if you want to have references to research being done with our software previously. We can input lots of different kinds of data types into Omics Explorer, and basically any kind of data which is expressed as large matrices of data is possible to import. So basically anything you can open in Excel or paste into Excel, you can also load into Omics Explorer. Initially, the software was developed for array data, but basically any sort of tabular data can be input to it. Also, we directly support lots of different file formats, for example, BAM files for RNA sequencing, cell and chip files from the Affymetrix gene chips, Agilent text files, and so on. Also, we have something we call the data import wizard, which allows you to tailor the input of any kind of data that you have in a table. So basically, any multivariate data which is in a tabular format works fine with our software. That's what I wanted to say. Also, the software is basically built for visualization and exploration, but you can do a lot more than that. You can do advanced statistical analysis with ANOVA tests, t-tests, regression analysis. And if you want to do very advanced statistics, customized statistics, we even have an open API to R, so you can actually extend the statistics analysis within Omics Explorer with your own R code, which makes it easy for you to tailor the statistical analysis to advanced experiments. We also have machine learning built into the tool. I'm not going to show that today, but I just wanted to mention that you can build classifiers, you can do classification, and you can use classification for prediction of data. When you have like a lot of samples of known character and you have incoming samples of that you don't know what group they belong to, you can use these classifiers to predict what group they should belong to among your experimental data. I'll also mention that there is an NGS module which allows you to browse a genome, view gene fusions in circle plots, and use the same RNA sequencing data for example, viewing expression levels as viewing genome. I'm not going to show that today. I just want to mention it because this is a second offering that we have on top of the regular product. So now to today's main topic, the demonstration of the software. And what I'm going to show is just a few basic steps and a few basic plots that can be brought up in Omics Explorer. So what I'm going to do now is to remove some of these extra windows that I have on my screen, which hopefully aren't seen on yours. But here, let's see here. Now, what we see in front of us is the main Omics Explorer window, which is where you end up when you are opening the program. So here's the main workspace. The big gray area in the middle is where your plots and your data shows up when you load it. And what I'm going to do today is to walk you through a couple of different plots and a few ways to manipulate them. So I'll open some example data here. A data set with leukemia data. This is child leukemia, about 150 patients, or to be exact, 132 patients. And we have array data from 22,000 genes here. So this is what you see when you open up a data set on your screen. This is the default view that you get when you start the program. It's called a PCA view, Principal Component Analysis. What you see here is each of these individual balls on the screen represents a patient. And this is the patient view or sample view. It's a three-dimensional plot. You can just click here and drag and move the mouse around, and in 3 dimensions, view the data set from various angles. This is very useful because when you do exploratory analysis and want to see what's going on, being able to turn things around and use your human vision to gauge distances and things like that, that can be very, very useful. As you see, it's very fast. We're using accelerated 3D graphics here, a standard feature on modern computers. So that was the sample view. We also are able to show a PCA plot of the variable view. This is your variables. And as you see, the cloud of variables here, 22,000 of them, moves around just about as fast as the sample view with just a few hundred, or just 130, sorry, different items. So let's go back to the sample plot here. Now, and I'll reset the settings here so we're back where we started. What you see on the screen is a principal component analysis plot, which is a way of visualizing how similar samples are. Two samples that are close by to each other. For example, these two samples here. If we turn the plot around, you see that they're close by in 3 dimensions, these two samples here. They are very similar. So if two samples are close to each other, they are similar in some way. If they're very far apart, let's take 2 extremes, this sample and that sample. They're very far apart, so they're very different. That's what the PCA plot shows. Now, we'll do some magic with the PCA plot. where we start doing filtering of our data. And we're going to do filtering by standard deviation. And what we're doing when we're filtering by standard deviation is that we're throwing away variables that contribute only noise. And we're keeping variables that contain information that cause these differences to appear. When we do that, the plot will change. Let's see what's happening here when I start doing filtering. I'll start doing filtering, and you see the plot is changing. And when I've filtered to about this point, something very interesting happens. As you see here, the data set splits into two very distinct parts. Those two very distinct parts you see here, they are two potential groups of patients that have interesting properties. Now, if I want to do some cheating here, because this data set contains annotations for the diagnosis posed by the doctors. This is acute lymphoblastic leukemia, which has two main types. So let's color these by the two main types and see what we found. There you go, B-ALL, T-ALL, the two main types of acute lymphoblastic leukemia. They could be found just by doing filtering on standard deviation. As a matter of fact, the B-ALL type has very many subtypes because it's a very complex disease, this one. And what we see here is that there is potential, for example, here for other groups. And if we color by these subtypes, we can see very clearly that we're starting to see an indication that we're finding the subtypes very easily just by doing this standard deviation filtering. I'm not going to go into detail today on how to discover groups in the data. I just wanted to show you the plot, but I also wanted to give you a context of what we're doing here. Let's go back to where we were, where we started. So where we started was just about here. Now, I can express my data in so many different ways. Different plots for different purposes. For example, the PCA plot is three-dimensional PCA plot is very powerful for human exploration and playing around with your data. However, it's not as good for publishing in a publication. If you want to do that, many publications usually ask for certain types of plots. And one type of plot which is very common in many publications today is heat maps. So let's go to a filtered down data set where we were about here, where we had found those two groups, and just plot a heat map on the screen instead. I'll move the statistics window here a bit so you can see. And what we see on the screen here is a heat map with our results. Now, if I want to zoom this out a bit, for example, if I want to squeeze in a bit more of the screen, maybe that was a little bit too much. Things get a little bit too small there. But if I want to squeeze in more of the variables, because you see variables are on the y-axis here, and on the x-axis we have samples. And I can scroll the heat map around like so. What we see here is potentially a group. You see the band of different colors here. And that's what we see in the PCA plot, because we're seeing the same thing as we saw there. We're seeing a distinction between a main group and an outlier group, the two different types of leukemia. The plots can be colored. If I want to do color leukemia type here, you see I get the same, this band that is sort of visible here was the subtype of leukemia that I've seen before. You can do more things to the heat map. You can order your samples by hierarchical clustering, for example, which is one way of expressing similarity in clusters. That was the PCA plot. That was the heat map. Let's take a look at a few other plots while we're at it. We'll go back to the PCA view where we started. We'll start over again from where we were at the beginning. Now, let's do a statistical experiment here. Let's do a two-group comparison, and we'll do a two-way two-group comparison, which is a two-sided t-test. The two-sided t-test we do on the leukemia subtype, and what we do it on is this one, E2A-PBX1, which is one of the subtypes of leukemia, and I'll explain why I'll use this one soon. This slider here changes my p-value, and as my p-value changes, the result of the statistical test changes as well. And here we go. Here we have two distinct groups. Those two distinct groups separate well in the plot, which means that they are very different. As I said, two samples that are close are similar. Two samples that are far apart are different. So here we have two groups of samples that distinguish themselves with a very good p-value. I have 2.52 times 10 to minus 23. So that is a really, really small p-value. I also got a good q value there. So here we go. My statistical test has found something interesting. Again, I'll do the cheating and look at the diagnosis. And if I look at leukemia subtype here, you see that the red group, which happens to be E2A-PBX1, is this one. So with a very high probability, I have found two distinct groups, right? But now to the interesting thing, because I've filtered down, if you look here, 25 variables are left in my dataset. 25 variables are left in my dataset, which means that these 25 variables distinguish these groups very, very well. I'll click on the list of variables here and bring up the gene symbols corresponding to those variables. And I'll have to move the window a bit so I get some more space here. I'll also bring up the p-value here. There we go. I'll even do. So here we go. If we look at the three variables that have the best p-value in the group, they all represent, because I have three variables representing the same gene symbol, because these are three different probe sets measuring the same thing, right? So these are three probe sets on my chip array that measure the same thing, and all three indicate this gene, which is kind of interesting, because this gene happens to be the gene that distinguishes this group from the others, right? This is the gene whose activity determines that this subtype of leukemia is active. Now, if I want to visualize this statistical test in another way, I can do the following. I can do a line plot where I select to have on my x-axis the active samples, and on my y-axis variables. So here we go. And I'll actually put in no, I'll do this instead. Sorry. Here we go. What we see in the line plot here now is I have a very big spike here. Now the x-axis is a little bit blurred because we have quite a few samples in the plot here. But what we see here is a very big spike for those samples that contain the PBX1. So these are our group that we were looking for. I can also change my plot. I'll show something that is much better to visualize this. I'll show a bar plot where I'll use the leukemia subtype and I'll select a variable. I'll select this one. There you go. I can also view that as a box plot if I want to, which is even better because this will show me much more clearly what I'm looking for. So I'll do a box plot where, no, not sample, I'll use, sorry, leukemia subtype. And I'll select a variable. I'll select one of the PBX. There we go. There's your upregulation of the PBX gene. And also, it shows me for each of the different groups in leukemia subtype which genes are different here. So here I get a box plot of that for the PBX1 gene, sorry. And that's just a few of the plots that we have. Data can be expressed in so many different ways. One of the most convenient ones, if we go back to where we started, is the PCA plot, because that allows me to manipulate things in 3 dimensions and view the data as an interactive 3-dimensional plot. The line plot is useful for publications, of course. So are scatter plots and bar plots. The box plot is useful to illustrate statistical relationships, with the whiskers here showing you your intervals for the statistical data. And of course, we support many different kinds of plots. These are just a few of them. You can even view your data as boring old numbers if you want to, but this is rarely interesting to most people. However, one neat thing that we have here is, of course, we support copying to clipboards. So there you go, image copied to clipboard. Oops, I was supposed to trying to open MS Paint. There we go. And I just paste my image to Paint there, or to a PowerPoint presentation or whatever. There you go. That's just a few features of the plots in Qlucore Omics Explorer that I wanted to show you today. Now, to your questions, if there's anyone who's got questions, let's do like this. There's many people here. So there's many people here, so I can't have everyone's audio on. So let's do the following. If you have a question, type it in the chat window, and I'll read it aloud to everyone and try to answer the question regarding the program. If you got any questions on what I showed you today. Is there anyone? Okay, again, if anyone has a question, please post the question in the chat window. No one? Is everything clear what I showed today? I want to mention a few more things. I showed you a little bit of the statistics. I only showed you a very, very small part of the statistics, because I showed you a t-test. We can also do a multi-group test. We can also do rank regression and other types of statistical tests. And I did mention during the initial presentation that we also support your own statistics in R, where you can extend the statistics available in the program with your own code. Then it becomes a little bit less interactive, because to recalculate the statistics, you'll actually have to wait for R to process it. But when you have calculated it, you can do filtering on it, like I did with the standard deviation filtering, for example, or with the p-value filtering. You can do that interactively. But when you recalculate the result, it takes a little bit of time. All right. As nobody seems to have any questions, think we call it a day for today, or maybe I should show you one more plot, because this is a plot that I've gotten a few questions about lately. If we go to the special plots here, there are a bunch of interesting plots that are useful to advanced users. We support Kaplan-Meier plots, Venn diagrams. Venn diagrams are really useful. There's going to be a webinar about specifically about those next week, and they are particularly useful when you're manipulating lists of variables. Here I got a question for someone. How do I manage zero values in my file? I suppose you are asking when you have like a value which is zero or a value which does not exist. Is that what you're asking? The person who asked that question? Okay, that's a good question. We support missing values. And when a value is missing, that means that the value is not there. For example, you have failed to read the data, or there is no valid data for one point in the matrix. We do support that. And when it comes to that, we actually do something called missing value reconstruction. And when you have missing values in a matrix, you have to reconstruct those values to fill in the gaps so the statistics will work and the calculations will work. And we support two different ways of doing that. One simple one, which is simple averaging, which is basically a simple, just an average. We also support an algorithm called K nearest neighbors. Okay, I hope that was an explanation of what you wanted. Otherwise, just tell me again. Here's another question for someone else. I want to know if you apply variance and optimization. You want to know how to apply variance and optimization in statistics tab. Okay. Let's go back to a PCA plot, because that's the easiest one to show that with. If we have our PCA plot here, let's reset everything to where we were from the beginning, and let's remove the coloring as well so we don't get confused by that. Oops. Color it solid. We also reset the view. Now, the statistics tab here has three, the statistics window has three tabs. One is called variance, where you can do variance filtering. Statistics, which can do statistical tests, and one called extended, which we'll leave for now because that's an advanced topic. That's the R interface basically. Here we have three different ways of working with the data set, but I'll show you the statistics and variance once more. The variance tab is really simple, because basically that throws away variables by standard deviation, and I can just move this slider here to do that. However, the attendant here asked for optimization, and that's a very good question, because we have something called optimize here, an optimize button. Find optimal variance filter based on projection score, it says on the help here. And this here happens to be the projection score. The projection score is an invention that we did here at Qlucore, a mathematical invention which is used to give you a guidance, a rule of thumb regarding variance filtering. Because variance filtering is rather difficult. If I start doing variance filtering here, at a certain point in time here, about here, it becomes very evident that I have two groups. But I can keep doing this. And the group is still there, and it's still there, and it's still there. But now I've thrown away too much data, so now the data degenerates. And if I keep doing the variance filtering until I'm at the extreme stage here, I've got 4 variables left, and now I have 2 groups again. But those are not the 2 same groups. Now, basically, what I've done when I've got only 4 variables left is that I've thrown away so much of the data that only the strongest signal in the data is there. And now, what happens to be the strongest signal in genetic data? Well, if let's just put in the, do I have the chromosome data here? Yeah, chromosomal location. Now, if we look at this is rather fun because two of these are the same symbol, XIST. That's actually the gene that determines one of the strongest signals determining the sex. So what we're seeing here is basically the boys and the girls. Now, that was a sidestep on what I was going to tell you. We were going to talk about the optimization because it became rather evident that if I do too much filtering here, my data degenerates to garbage. And as you see, if you look at the projection score here, that one actually varies while I'm moving the slider here. So here it goes down after a while. It becomes evident that it goes down, becomes yellow, and here it goes up again. And then here is the optimal mark. If we keep going, it goes down again as the data deteriorates into this again. Now, the optimization button actually tries to find the maximum projection score, to find that optimal point where you see the most in 3 dimensions. The projection score is just a guideline. It's a tool to help you. If you want to read more about the projection score, there's plenty of that. We've got white papers on it on our homepage, and also in the help in the software. There's also references to the scientific papers. The question now, is it necessary to do this? No, but again, the projection score gives you a statistical tool to help you with this. Someone else asks here, we apply 0.2 variance for mRNA data. Do you think it's necessary to do it, or do we just keep it to 0 and look at the data? Well, I can't give you a guideline for your data, what is the best standard deviation filtering, because this will help you throw away the noise in your data, and that depends on how noisy your data is. It can be very noisy due to experimental conditions, due to the type of data you're measuring, due to equipment. It can be all sorts of things. So I can't give you guidelines on if 0.2 is enough for your data, unfortunately. What I do tell you here is that we offer this optimization to try to give you the best projection score, and the projection score is a guideline on where the variance filtering is supposedly giving you the most information. Now, it's also because there is a peer-reviewed paper describing this, which is referenced in our help in the software, and then you have a reference to why you did that variance filtering, because you see very often in scientific papers, we did variance filtering on our data and we stopped filtering at this or that value. But then you can immediately criticize that by saying, okay, why did you stop at that particular value? Why didn't you choose this one instead, where you could have seen that? So the projection score, since it's a peer-reviewed publication, will be useful in those situations where you want to have a scientific reference to ask you why you chose this particular level of standard deviation filtering or variance filtering. Right, that's what we had time for today. I hope it's been informational and that you learned a little bit. For those of you who haven't tried the software yet, there's a free trial of the software. You can download it from our webpage, and you just register on the homepage, download the software, and you get a 10 days free trial. The free trial is basically limitless, except that it cannot export plots to the disk, and you cannot copy things to the clipboard and things like that, because you're not allowed to use it for publications. You cannot just download the software, use it for 10 days, and publish something. We don't allow that, but we allow you to try out everything in the software. And the automatic trial you get is for the basic software. If you're interested in the NGS features, you'll have to contact one of our sales team members and ask them for a specific trial for NGS. Okay, thank you very much for today. It's been fun. and I always like to show this to new customers and people who are interested to it. If you're interested in some more topics, I can tell you that there is a webinar already in a week. And as I said, that's going to be specifically about Venn diagrams and how to manipulate lists of variables in the software to combine them and refine your searches by doing that. I can recommend that one. Otherwise, thank you for today, and thank you for listening, and see you on the next webinar. Thank you.